yeah, that's true. What did I do? I didn't touch anything.
Hi, Amy, how are you?
Hi, trying to save a baby bull. How are you?
Oh, okay. Trying to save the baby boy. He's sick.
Yeah, good.
What's wrong?
Got a Con Ed and doing it while I'm flying.
Sorry, I'm 10 minutes late.
No worries. We're just waiting on you.
Okay. Yay then.
First time. Everybody's got packet, everybody's got everything, right?
Right.
Okay. Okay, so let's get started. We're on our second to last class. We're going to talk about insects next week and we're done.
Oh.
And as far as the future of this class, we're looking to restart it the first week of July or the second week of July if my business plan development training class doesn't go off.
So that.
And I won't know till the end of the week. So by next week, I will be able to tell you if we're going to run this class or business plan development training for farmers next.
I want that, the business.
That's a good class. And we're going to do it in Pahoa and in Hilo at the same time.
Oh, wow.
I'm going to teach both.
Who will teach Pahoa?
Me.
And Hilo.
At the same
Yeah,
time?
Yeah. So I live right there. It's easier for me. They're good. We're going to have it at Ku'okala, most likely.
Oh, you're already down there. Who eats?
No.
Choke mosquitoes down there. Choke mosquitoes.
It's my house, man. And fire ants and everything. Alright guys, so we're going to talk about plant-associated microbes today. I do apologize, as you can see there are no speakers up here because I didn't find any videos because I spent all my time finding out real information, so it's going to be one of those droning on lectures without exciting colors and all that. No, the pictures are actually very cool, so I'll go through the pictures and what I've chosen to present to you. So if you see me skip over a photo, just say, James, what's with the photo? And I can go into it because for the most part they pertain. We're going to talk today about two things. We're talking about two things today on my list. We're talking about plant beneficial microorganisms, and we're going to talk about plant pathogenic microorganisms. So, when I refer to any other science of plants, like botany or morphology or soil science, I want you to know I'm only focusing on the microbial plant association side today. So I know if you see me steering off outside the plant area, outside the rhizosphere, just say, James, get back in the rhizosphere and focus on this, okay? So that's the goal, is to focus on plant beneficial microorganisms. We're going to talk about that for maybe an hour, and then the rest of the time we're talking about the pathogens, how they do it, why they do it, and which ones are doing it and why, for the most part. Okay? So right here in the picture, of course, we have what? Mycorrhizae.
But that's not accurate though, right?
It's very accurate.
Well, but this is such a tiny plant and it's got such a giant root system.
Yes.
With mycorrhizae on
It's a legume also, and it has nodules as well. This is a lab created for display type thing. And if you think about it closely, it's actually two panes of glass, probably about this wide. So the roots actually go down pretty far because it's not going out in a ball, it's going flat on a plane. But yeah, so, because you can see it comes down and it wants to go anywhere, but it's trying to go any direction but down, but it gets steered downwards. But anyway, so yeah, we got the mycorrhizae being a fungi that has close associations with plants. We'll get into that. So for the most part, the next hour we're going to talk about plant beneficial microbes. Diazotrophic bacteria are one type of plant beneficial microbe. Those, of course, are going to be the nitrogen fixers. Okay, so these can be free living outside the plants or endophytically on the inside of plants. And the inside ones, of course, have a very close association with those plants. And the ones on the outside sometimes have associations with plants, and some of them are just cruising along doing their own thing, making their nitrogen for themselves, and they happen to dribble some out onto the plant surface, root surface that is. Plant beneficial microbes also include biological control agents. So several bacteria are in that category where they do things to control their environment. And then we also have plant growth promoting rhizobacteria. Plant growth promoting meaning it's going to stimulate some sort of growth or defense mechanism or enzyme production or hormone production, like I said, in the plant. And then also there's fungi that do the same thing, most of them being mycorrhizas.
Which plants are these?
Which plants are what, where?
Like either one, like example for one, like if I wanna promote more of the rhizobacteria.
Okay, so almost every plant except for like brassicas and a couple plants, families, being the lettuce family, cabbages and that kind of thing. Almost every plant has some sort of mycorrhizal relationship. Sometimes it can be species specific, sometimes it's general, but for the most part we found that every plant has the ability to have that sort of relationship. Now, only certain plants can fix nitrogen internally with the help of microorganisms, and of course some bacteria will do it just outside the plant. Biological control agents, that's going to be microorganisms that are doing that to plants or to other microorganisms. Plant growth promoting bacteria, yeah, so that's a relationship between the bacteria and the plant. Okay, did I clarify that good enough? Okay, and of course in this picture, this is going to be a root nodule, microscopically root nodule, where we have nitrogen fixing bacteria contained within. It's actually just tightly packed in there, and they're cycling through their life cycles and making metabolic substrates and putting them into the plant, and then some of them are being secreted out into the environment.
So these are the pink ones, I just have to ask them.
What? What is the pink one?
No.
These are going to be, oh, you're talking about colors, yes.
Yeah, actually.
You're cracking jokes already, we just started.
That's the mochi. That's the mochi.
Mochi.
So the pink ones would be, the mochi ones would be the nitrogen fixers. Okay,
Thing.
alright. It's very difficult to determine how these microbes improve plant health. That is because, first of all, we don't know a lot about them. Second of all, they work together, and it becomes this super complex equation at that point, which you start bringing in a lot of different kinds of microorganisms, kind of doing their thing to one plant. It's kind of hard to tell who's having what effect where. And several factors must be considered, basically the production of metabolites related to root development, growth, and pathogen control. Metabolites are what again?
Energy being used.
Usually by a microorganism. And so if you are creating enzymes, that's a metabolite. If you are fixing nitrogen, you're doing a metabolic activity, some sort of, it's all the same thing, okay? So these are like the functions in which the bacteria do their thing. Those things are related to, so the bacteria are related to root development, growth, and pathogen control. And that's the beneficial side, what they do to help out plants. Difficulty determining direct effects on a specific total activities of the microbes. Like I said, it's very difficult to figure out what is doing what, and what is the direct effect, or is it an indirect effect of these metabolic activities or combinations. The indirect effects due to the enhanced availability of nutrients and growth regulators. If there are new nutrients available, usually coming from plant roots, roots of course make exudates. They make all kinds of juices that they send out, their chemical signals, their hormones, their sugars, all different kinds of combinations coming out, indirect effects due to enhanced availability of nutrients, and then growth regulators being hormones. Okay? So they play in a lot with the hormone activity. It's hard to tell if it's the plant making the hormone or is it a microorganism making the hormone and giving it to the plant, because microorganisms have the ability to reproduce or make their own plant hormones and then pass them on to plants and allow them to make them do what they want based upon the hormones that they're giving the plant. Does that make sense? They're actually able to control and drive what a plant does because they're able to make these hormones and give them to the plant. Remember that all they're doing is creating a molecule. They're creating a molecule, and the plant sees it as a molecule, whether it makes it itself or if it's made synthetically, but outside the plant. Just like a multivitamin, the human body really doesn't notice the difference. You're taking calcium in, it's calcium, it gets absorbed. It's the same way. It's consuming a molecule. It's the same concept.
What kind of hormones?
Gibberellic acid, cytokinin, we actually will go through them later. There's about, I think, 9 of them now. We keep finding more. Let's talk a little bit about plant microbe interactions. Okay, so their soil-borne microbes interact with plant roots at the root-soil interface. As I just said, a lot of interaction goes on at that root zone. So when I talk about soil microorganisms in 2 classes ago, or last class, these are going to be microorganisms just around that root zone. Rooted exudates and plant decaying material provide source for carbon compounds for heterotrophic bacteria. So root exudates, the juices and sugars that come out of the roots, and plant decaying material, so leaf matter that is falling off in dead plants, they provide, of course, carbon sources for the biota, usually being saprotrophic, right? So the ones that break down organic material. All right, let's talk a little bit about the 2 zones, so we can get a little. We're starting on vocabulary today. It's like almost all vocabulary, but it's just understanding the concepts behind what we got. All right, so we're talking about the rhizosphere and the rhizoplane. Okay, so the rhizosphere is the zone in the soil that is really close to the roots. So it's a soil environment, think of it as a soil environment that is really close and being heavily influenced by the roots. Rhizoplane is the actual root surface. Think of it as this is the plant-based side. So there's 2 ways to look at that area. It's because there's things going in from the soil to the plant and things coming out from the plant to the soil. You got to look at it as both ways. So we call it 2 different things, right? Rhizosphere, rhizoplane. We have, of course, plant exudates, as I said. These are generally leaked out of the rhizoplane, out of the rhizoplane, which is, like I said, just the surface of the root. Usually leaked out because there's this zone of a lot of cell division. Remember, this is the root tip. This is where you got the parenchyma cells that are developing into structural defined cells. So you got a lot of juices coming out and leaking. And then, of course, we have secretions that a plant will purposely release into the environment and secrete into the environment. Lysates is basically when cells are burst or exploded by osmotic pressure. It's usually the cells in the furthest, in the outside of the root. And so this is what happens during growth. There is this kind of outer layer of cells that they know are going to burst and pop. So they're going to release juices as well, right? and it's kind of part of growth, so it's understood. We have the same thing on our skin. You know, as skin develops, there's a layer of cells that just kind of are exposed to the environment, and they burst and flake off, and so we have the same kind of epidermal layers. And then of course we have plant mucilage. These are polysaccharides. These are proteins. It's usually going to be right around the root tip, and it is a form of lubrication for the plant as it pushes the root tip through the soil. And we talked a little bit about that on plant physiology today.
So the green things are what coming out as
I'm not even gonna answer that question. It says right here. But anyway, so you have plant mucilage is right here, and it can, remember this is a aquatic zone, there's water all out here. So the mucilage is kind of just streaming out into the environment. Okay? Here's a really nice diagram of what I'm talking about. So the yellow line being rhizoplane, just the area that is exposed to the soil on the plant side. And then the rhizosphere, they define it as including the inside of the root, endorhizosphere, and then the ectorhizosphere. I don't know if I would define it that way. It's really more defined, like I said, right up to the root surface in the soil. So on that same concept where there's juices and sugars and polysaccharides coming out, it's known that as seeds germinate, a large amount of carbon and nitrogen compounds are released into the soil. So imagine a corn seed, it comes out, it's got that big stalk that comes out, and that thing is just loaded with those sticky, oozy sugars and polysaccharides. Also includes, like I said, sugars, organic acids, amino acids. We were talking about that in the last class, amino acids being very heavy in nitrogen. And then vitamins as well. Induces vigorous competition between microbes at this point. So as a seed pops and germinates, finally it's setting that root into the soil, the microorganisms are becoming incredibly competitive for that root zone. So as the roots are growing, as the cells are dividing and becoming certain structures, those microorganisms are waiting to see what structure it will become, and then boom, get right in there. As soon as the structure is formed, the cell structure is formed, not even the structure itself, but the cell structure of that area. So if it's a root hair, something like that, epidermal cell, it will wait for the root hair to come out, and as soon as the root hair comes out, boom, it'll hit it. But before that, there's another microorganism that is on that epidermal cell before the root hair starts to elongate. And then there's different exudate comes out, so there's a new one that comes in that wants that. And they may intermingle, and they kind of, the more exudates, the more things that are coming out of the plant, the higher the population and diversity of microorganisms.
Well, they've got all these commercial products in the bags, gardening soil, things like that, but I remember you saying before that they purify it or whatever, you know.
Yeah.
So there's not really much.
No, it's inoculated with what they inoculate it with. So, I mean, like, ones like Fox Farm and that kind of thing, they put vermicompost in there, which is incredibly heavy in all these beneficial microorganisms.
So most of the products would have those microorganisms?
Yeah, they pretty much make sure that they have those in there.
Okay.
You know, they have some sort of either synthetic or organic source for those, and then they put them in there. But like the ones you had, that wasn't organic. It was a mycorrhizae inoculated soil, and what they had done is like I said, they cleaned it, only put that mycorrhizae in, and it's so dominant in the area that no other microorganism is going to have any foothold. Right?
Mhm.
Okay. No, it's not. So I tend to get
Yeah,
the
part
art.
because it says it's got mycorrhizae.
Yeah, it's got mycorrhizae, but what is it? What does mycorrhizae do? It just absorbs nutrients that you pump into it. So wouldn't you rather have something that has nutrients available already in it? That's not anyway. So yeah, I'm not going to advocate one company or the other, but I pretty much use Fox Farm for the most part.
Oh, you
The
do?
Ocean Forest Floor one. Microbes, of course, can differ between plant species, or they can. Some of these microorganisms can just jump from one host to the next. But for the most part, there is this kind of relationship between certain species and certain plants.
So that's when people plant like companion plant together, that's because of that kind of theory? Like put whatever microorganism will be together because it's similar plants?
I would hope to think that they would think that deeply about it.
Oh,
But a lot of times it's, is this plant shading my plant? Is this plant protecting my plant? Is it bringing beneficial insects?
right.
So it's like
Oh,
very rare
okay.
that they'll think that deep into it, but some of the guys do, so yeah, those that do, more power to you. Okay. Beneficial microbes, of course, can be biological control agents, as I mentioned earlier, and growth promoters. I say that because a lot of times one microbe does both activities. So biological control, making the environment suitable for itself and the plant, and then growth promoters is stimulating growth in the plant.
As a byproduct, kind of.
No, as a purpose to create. So if a microorganism like Bacillus subtilis, who loves to hang out around the root zone, was to excrete some sort of enzyme or hormone that caused the plant to grow more roots, it wouldn't be like kicking money up to build some more condos down below. It's kind of like what they're doing. So if they say, hey, we need more roots down here, it really benefits them, so they're going to do that. And it helps the plant. Okay? Well, of course, there's going to be different zones of metabolic activity. Metabolic activity, of course, being, we went through that already. Nobody's confused about metabolic activity, right? Okay. It's always a laugh, right? Exophytically. What does that mean? Outside where?
The organism,
What organism?
the one we're looking at. The green one.
These are all true statements. Just say what it is. Phytic means plant. Phytic means plant. So, exophytic. Matt, how are you?
Good.
Grab a thing, grab a thing, grab a thing. Sign in. We are on which slide?
4.
The
Page
fourth,
4.
page 4? Page 4.
Second.
And we're talking about zones of metabolic activity. These are going to be areas on which you will find microorganisms doing stuff. It's basically as simple as I can put it. And we're trying to figure out what exophytically means. We figured out exo is the outside and phytically means what? Plant, outside the plant. So a lot of metabolic activity happens on the outside of the plant. Here's an example, a leaf with bacteria on it, right? Outside the plant, on the leaf versus in the xylem, the phloem, or inside the root structure, right? The vast majority of known soil and plant beneficial microorganisms are this type of beneficial microorganism. Actually, most microorganisms in general are going to be this kind. I have an asterisk there. Why?
Something this word.
We don't know. We have no idea. We've been looking at endophytic, inside the plant microorganisms, for like 10 years because only till the point where we had DNA, the ability to do DNA sequencing, could we figure out what was even inside. So, yeah, it's a bold statement, isn't it? We know about 1 to 5% of microorganisms on this planet. It's tough to say what is doing what where, right? So they generally, for the most part, the exophytic microorganisms generally originate in the soil. So they do their thing in the soil, and then they end up on the outside of a plant to do its metabolic activity. So its purpose in life is to get to that zone of that plant after it left the soil and do that specific thing. Which is why life is so amazing, because you find all these things that, these microorganisms that their whole sole purpose in life is to kill your tomatoes. Bastards. Right?
But you can't blame them because you don't know what they actually do.
What I can do is look at humans and just say that those things are doing nothing compared to what we're doing. So we are the worst things to ever hit this planet. So
We are.
I can never look down on any organism,
Right.
ever,
True.
because we are POSs. So anyway.
Oh, I got it, sorry.
We are
I just
positive.
learned what my.
Positive.
I'm so slow.
Alright, so other zones of metabolic activity, excuse me. Can you make sure, Matt, that air conditioning is turned to like 2 or 3 on your way in or out? I appreciate it, brother. He was like trying to pee. Alright. Endophytically means what?
Inside.
Yay, within the plant. And usually these are obligate endophytes. Some of them are facultative endophytes. What the heck does that mean? Obligate endophytes. Wait,
That there are specific,
no idea.
functions.
No.
So these are going to be ones that have to live on the inside, do most of their reproduction and their life cycle on the inside of plants.
Yeah, that's what I said.
Yes.
You did not.
That was just what I
Yeah,
said. They're
yeah.
obligated to do
Yeah.
only
It
specific
really
things
speaks
Yeah.
it.
within the plant.
And then facultative
And then
can
they're-
live in and outside the plant. Now, okay, they all can live in or outside inside the plant, but they can do metabolic activity inside or outside the plant. A lot of them can only do metabolic activity on the inside of the plant, and then they leave that plant and they run around trying to find another one so they can go inside and do their metabolic activity. And if they don't find someone to do that, then they don't live. Okay? So facultative can just, I'm out of here. Boom. Jump ship, chill out in the soil, wait for something to come along or not. I'm cool. I don't really need to be anywhere. Facultative. That's describes me. I'm very facultative. Modulation by altering ethylene levels. They are able to modify the inside of the plant by altering ethylene production, which is another one of those hormones I was trying to describe. Okay, so inside the plant, they alter some juices and some hormones, and they can make an environment that works for them. But remember, most of them are on the outside of plants.
Right.
So we have some wonderful hyphae on the inside of cells. I've used this slide before. I just like the color blue.
Yeah.
Look at this one, Nona.
Really nice orange coloring, right? Actually what that is a great picture of a stem. And what they did was that fluorescence, that fish technology where they put the fluorescence on the pathogen. And so the stem cells are red and the pathogen is yellow, glowing yellow. So it shows the pathogen on the inside. And if you've ever seen, and I wish I would have had more time to do videos for you guys, even though I never would have gotten to them. Check out some of this fish technology, how they're able to look on the inside, the guts of like worms and stuff.
Wow.
They don't even need to cut open the worm. The worm can just cruise around, and if they want to see if a certain microorganism is consumed in the environment and then camps out in the stomach of the worm, they'll just give it fluorescence, wait for the worm to eat it, and then the gut of the worm glows, and it forever and ever glows. We know that it sticks around in there, or does he poop it out? Or does he not eat it, you know, and so it's really interesting. But with the plants, the plant stuff is amazing. Because sometimes they'll change like the gene for the stem and make it glow. So like the whole plant, if you turn the lights off, the stems glow blue or something. It's absolutely amazing. And it's great because it's nothing, all they're doing is taking a gene out of a fluorescent bacteria and putting it in another bacteria or a fungi. And
So what is it like if I want to search on YouTube?
FISH, fluorescence.
F-I.
You're making me look it up.
Fish.
Anyway, it's fish.
Okay.
We call it fish technology, fish observation, microscopy, whatever. Anyway, so endophytically you have different kinds of microorganisms that we categorize in 3 different categories. There's competent, opportunistic, and passenger. Competent being they possess key genetic machinery to colonize the endosphere. That's the inside, right? The environment on the inside. So they possess key genetic machinery. So they can produce, they have genes that allow them to produce enzymes or have genes that has, for the fungi, could have a poker. They can poke a hole in the cell and get on the inside. So this is very competent. This is somebody that was genetically evolved to do this activity, to get inside this plant. There's opportunistic. Coincidentally encountering root tissue. I like to think of this guy just cruising along, minding his own business, and all of a sudden, hey, there's some roots for a plant that I really could go inside and reproduce. That sounds great, let's do that. So they go inside and they do their thing. So they're not necessarily, so if it was a bacteria, right? Bacteria can't poke a hole in a plant. It can't. It can create an enzyme that can kill a cell, but the next cell still has a cell wall. It'll have to kill that cell wall. It can't like poke a hole through the next cell wall. Like fungi can come in and poke holes in cells. So if you were, where was I going with that? I was saying something, coincidentally encountering, and if they was opportunistic, where was I going with that? Come on, James, think.
It's easy to go to the root tissue, but with the
Oh!
cell.
I remember. Okay, here we go. You're cruising along, and you can't get inside the root because you're a bacteria. But if somebody came along and with a spade and cut the root, and you're opportunistic, and you're cruising along, and you're like, hey, there's a hole in the root, and you go inside. And that's going to be opportunistic. So if there's a broken root, or if there's a stem that got broken off, or somebody pruned,
Right.
and then boom, ready to go.
Just like today, I was cutting the sides of all this mound and dirt and stuff like that, and I cut it for myself and everything, and this truck kept coming by, and then he wouldn't dig nothing until I got back and started cutting, and he asked me, well, could you do?
So you waited till you loosened everything up.
Yeah. I was gonna help him out anyway, you know. But
Yeah, right.
Yeah, but you know, that's an opportunist. Huh?
Totally.
He kept going back and forth until he seen me come by. You know?
Weasel.
Passenger is more like chance events. This is literally an endophytic fungi spore is swirling through the air. At the same time, a dog runs by your tomato plant and breaks off a branch. This is like literally by chance. This is it won't happen again in a million years. Guy cruises around, lands right on the spot that the branch broke off, and now the fungi inoculates that. And actually, I don't say it never happens because gray mold basically survives that way. And we'll talk about gray mold later. So, but yeah, chance events, kind of gray mold. What gray mold does, it sends out so many spores that it's going to get a chance event. And it's able to sustain itself by putting out so many propagators.
It's mostly parasitic.
Yeah, totally. And I hate that microorganism. Totally wipes out a lot of my crops. Effects of plant beneficial microbes. Indirect passive effect, positive effect, excuse me, or a direct positive effect. We'll go through that. Here's a great example of Bacillus subtilis, as I mentioned, the microorganism that loves to hang out around root zones and is an antifungal, antibacterial for the outside of your root zones. You can actually buy inoculant, which I use heavily, and use it on your root zone and make sure that no fungi get into your plant. It's about $100 a gallon.
What's
It's like-
which one?
It's like all those logs from Columbia, the Columbian River.
Oh, yeah, we go another river, yeah. And you can buy it at the greenhouse store. There's one called Cease, C-E-A-S-E. Actually, it's made here. Somebody over on Railroad, was it called Railroad Road?
Railroad Avenue.
Avenue? Somebody over there's making it. Anyway, so of course we have indirect positive effect and we have direct positive effect. Indirect will basically adversely affecting the population density, dynamics, and metabolic activities of plant pathogens. So it's active, oh, I'm sorry. Okay, so it adversely affects those populations, but it does it because it's trying to do its own thing. So it's trying to find a nutrient source, and in trying to find a nutrient source, it creates antibiotic compounds, releases them into the environment, and then finds that nutrient source. Or if there's a microorganism that isn't affected by antibiotics, and there's one sending out antibiotics, and they're both going towards the same nutrient source as competition, yeah? Lysis as well. That's the osmotic exploding of the cells. This releases, well, it's actually lysis on the plant pathogens. So microorganisms can create osmotic conditions that will pop the cells of plant pathogens. Does that make sense? So if it's a bacteria, it can create osmotic conditions so that the whole cell wall eventually pops. It does it by changing concentrations of salts and other wonderful metabolic activities. If you can think of it, they can do it. There's someone out there that can do it. It's really amazing. I like to think of them as superheroes, the beneficials, because they really have an uphill battle. I mean, there's so many out there that are either benign or pathogenic. Hyperparasitism, I've talked about this in class before. Actually, that wasn't this class, it was natural farming. I talked about it with lactic acid bacteria. Do you remember, Gus, with hyperparasitism? The reason why lactic acid bacteria works. Why does lactic acid work?
Well, it works on algae, right?
Well, it works on any nutrient source.
Yeah, well, it takes away their source.
Right, so it'll actually allow the pathogen to create a hole or a nutrient source and then fill that hole and cause the plant to build up its defenses or to fight against a pathogen even though it's not a pathogen,
You
it's
can
more of
explain
a...
it like that.
I don't know how else to explain it. So many different ways. Hyperparasitism. Everybody got that?
Yep.
Chasing out another one's nutrient source. We have competition. This is an indirect positive effect. Of course, competition, including space. Things get crowded down there, guys. Of course, there's the nutrients on the root surface. There's only so much square footage, square millimeters, right? So there's going to be a competition for that space on that root surface. Colonization of the rhizosphere, so just the general area around the root zone. All those wonderful mucilages and juices coming out. As they colonize these areas, there's kind of zones of concentration. Does that make sense? So in the soil, it's a water media, right? And as you get further away from the root surface, these exudates are at different concentrations, more diluted by water. And so if one microorganism would like to have this specific concentration, it will stay right on that area on the outside. Yeah, it makes sense, but the thing is, we're still talking about limited square footage. So just the root zone is limited square footage, and then as you work your way outwards, it doesn't get any bigger. It gets bigger, but there's still a limited area, right? So there's still competition. Successful establishment of the root zone. Essentially, a lot of microorganisms are able to anticipate where the root tip is going to go and can jump ahead of the train and start setting up shop waiting for the gravy train to come in. Okay. Another indirect positive effect of beneficial microorganisms is antibiosis. I mentioned this in the last class. In the soil, it happens a lot, but it happens in plant relationships as well. So a microorganism will protect a root zone or an area of the root with antibiotics. They will protect the stomata with antibiotics. They will protect any area where there's a hole. And also fruit. We'll talk about that a little bit later. Plants will inoculate the outside of fruit with antibiotic bacteria, which allow, antifungal as well, which allow the fruit to last longer. So if you have a really nice inoculation on your mango tree, your mangoes will last longer in storage and distribution. So there's a huge science behind that and all over the mango industry, trying to inoculate their plants with these antifungal bacteria and fungi. Okay, range of antimicrobial secondary metabolites, extracellular lytic enzymes. So in addition to antimicrobial, this also does this lytic thing where it'll create an enzyme that will change the osmotic pressure of a cell, a bacteria or fungi, and burst the cell. Any questions on that?
What are we looking at?
Good question. We are looking at Azospirillum, which is a antibiotic producing. That is not right. It is not Azospirillum. This is Penicillium. Yes, Penicillium. It's an antibiotic property in the soil. Also, Penicillium has a lot of pathogens as well. So don't think that family, that they're all going to be beneficial. So only specific species or organisms. Okay? So yeah, so antibiotics look like you got bacteria, right? And then zone all the way around where the sample was placed. Lysis, a little bit better description. It's when microbes burst due to osmotic conditions, as I mentioned. It's kind of like jumping on the grenade. Does that make sense? So if a pathogen comes in, there are certain microorganisms that have antimicrobial or antipathogen or enzymes in general that are in vesicles that are they're just storage units on the inside of the cell. And those can't come out until they burst. So they will actually change their osmotic pressure and purposefully burst to release these enzymes and materials into the environment to kill the pathogen that's going to hurt its host. Okay, so it's like, jumping on a grenade. It's coming to get us, I'll get it, boom. You're like in Star Wars, right? They always go up and they shoot down the comet or whatever.
So what's the trigger on that?
That is presence of the pathogen. Pathogen releases enzymes.
Right.
This specific microorganism will sense that enzyme production and start creating its own. Well, the genes will trigger a reaction for it to go into this mode. And I wish I had the video, sorry guys. But they'll show like a pathogen fungi coming in, and then these bacteria just coming at it and at it and at it, and then it eventually just dies because they can't handle the concentration of antimicrobial compounds. Okay?
Yeah, so in essence, it's a sensor, and then after that, it goes after that.
But all microorganisms are that way. They're microorganisms that their sensory devices are heightened at all times, waiting for some trigger to set something up. And they're not thinking about it because they don't have brains. What they do is they have DNA. And what you start doing is you look at the DNA structure. If this enzyme comes along, it will fit this switch, this switch, this switch, and this switch. And then those switches will cause the cell to do something. And it's a reaction to the presence of the pathogen. Any questions?
We're doing something similar to that up at the river, above the river, because we have a lot of kids downside enjoying the stream.
So you're just sending kids
No,
like
no, no.
at a million miles an hour?
And we're thinking about, now, flash flood can come anytime, even if the sky is nice.
Definitely.
So we're putting out sensors at different heights up on the river banks and stuff like that. I'm looking at about getting close to 10 of them. And then it's battery operated, so as soon as the water comes over and hits that sensor, that's a spring device, it trips it, and then it'll give a loud shriek, so the downside will be able to hear it.
That's awesome.
That's stuff we've been thinking about. But that's
It's exactly the same thing. You're hitting triggers, right? You're hitting switches. And it's kind of just, so you have a reaction to the change in the environment. Plant beneficial microbes' indirect positive effects includes hyperparasitism. This is very well documented in the Trichoderma species. So that's going to be a fungi. And it's when they're, so this is Pythium is a pathogenic fungi, right? And then the Trichoderma is this, can you see that?
Yeah.
like a tapeworm.
Yeah, like a tapeworm in there. They are able, these microorganisms are able to secrete chitinase and cellulase, which are both enzymes for breaking down chitin and cellulose. So by doing that, they can get inside chitin. Who has chitin?
The skin of the organism.
What organisms have chitin?
Bugs.
Yes. What else?
I shoot for one.
Fungi. Fungi have chitin. So it's able to get inside fungi, pathogenic fungi, and break down the cells on the outside and get on the inside of the pathogen. And then this will break down cellulose, which is what plant cells are made.
So the trichoderma is actually a parasite.
It is a parasite. It is a hyperparasite. It is a parasite on the inside of a parasite.
So that's kind of like what the pigs have, trichinosis. Yeah, it's a bacteria that if you get it inside your body, it can damage your liver, everything.
Sure. Well, yeah, the pig's going to be a vector. We'll talk a little bit about that in a little bit as well. So, this microorganism has to come in contact with the pathogen. Some of them can be fungi that have coiling hyphae that wrap around pathogenic hyphae. So instead of going on the inside and devouring the nutrients on the inside or doing whatever it's doing, it literally will just lasso up fungi and then send out enzymes and secrete out, or secrete in their, absorb in their nutrients. Enzymatic digestion of cell walls, so enzymes can go out so that cell walls can be broken down and digested. So you got to think of it that way. These guys aren't like comic book superheroes out to get the bad guy. They don't know what they're doing like that. They are looking for food. So this is how they get their food. They just happen to do it on really, really bad stuff. For us, as agriculturalists. And of course we got a lot of penetration. Keep it clean, Gino. Plant beneficial microbes. Direct positive effect on plants includes, so we just did indirect, so we're doing direct. Phytostimulation and biofertilization. Like I said, it's a vocabulary day. Let's go through those. After I take a sip. Phytostimulation is when a microorganism affects a plant by doing one of many things, including producing phytohormones. Like I said, microorganisms can create plant hormones. Put them inside a plant and make the plant do what those hormones are made to make the plant do. So if it sends in auxin, it will trigger the plant to grow. If it sends out ethylene, it will trigger the plant to flower. That kind of thing. Got that? Also, toxic compounds as well. And this is well, I guess if you want to change the morphology of a plant to be conducive to the environment on which the microorganism appreciates the most, it will actually kill off, some plant beneficial microorganisms will kill off certain areas of the plant to create a zone of reproduction or absorption of the nutrients. But at the same time, it's not there to kill the plant, it's just making a little home for itself. The production of plant growth promoting regulators. We're going to talk a little bit about that as well. Plant growth promoting regulators, PGPR for short, is described over there in the diagram, but we'll go through it afterwards. It's basically when microorganisms, beneficial microorganisms, these guys right here, they release metabolites, enzymes and such, antibiotics, also to help protect the symbiont being the plant, where they have a symbiotic relationship with, right? They produce enzymes, which have specific antagonism and also antibiotic properties to pathogens. Sometimes pathogens, we'll get to it later, but I'll bring it up just briefly right now. Sometimes these microorganisms can stimulate this plant to think it's infected by this microorganism to trigger a defense mechanisms. So we'll get into that later, but that's this ISR right here. And then of course there's infection. Sometimes the microorganism gets into the plant. But for the most part, you have this kind of relationship where the plant gets exudates, and exudates go up to the plant, and there's this communication and a nutrient source in between the plant, and then there's the pathogen here, and they all do what they can to combat the pathogen from entering. Phytohormones. So one of these exudates that comes from here that goes to the plant might be a phytohormone, might be gibberellic acid, whatever it needs to make sure that that plant is healthy enough so that it can do what it wants, or not what it wants, but do what it can to get nutrients, and that's all it cares about. Remember, the only thing anybody cares about in this equation is nutrients. They're not out to get anybody. Chemotaxis. This is kind of a broad spectrum idea for all biology, but it's basically the movement of concentration in response to chemical stimuli. These microorganisms can send out chemical stimuli and, like I said, make these microorganisms, these pathogens, think things, because not that they have brains, but think things that aren't true. This plant isn't the same host that it thought it was. Chemotaxis is that kind of thing. So it's sending out chemical signals, chemical stimuli, and causing a movement or a change in concentration of the pathogen. So, does that make sense? It's not really an antibiotic, it doesn't kill the pathogen. It just says, hey, there's nothing to see here. And then the guy goes, yeah, okay, cool, I believe you, and then takes off or whatever, right? That kind of thing.
Telling you, move on.
Yeah, keep moving, nothing to see here. Plant growth promoting regulators include nitrogen fixers, and these would be mostly going to be endophytic nitrogen fixers. What does that mean again? On the inside of the plant. And why would this be kind of a beneficial direct effect? It's because the nitrogen that's being fixed is given directly to the plant, right, in this situation. Where if it was free living nitrogen fixer, it would be doing it for its own good, right? Can consume various carbon sources, meaning pathogens, pathogens being, almost everything being made of carbon, right? If it could break down an area of carbon on the pathogen, when most of the pathogen is made of carbon, if you can break down any one of those molecules, it has a major effect on pathogens. There's incredibly fast reproduction with PGPRs. So typically PGPRs are going to be in the Bacillus family, which I'll get to later. But they're going to be usually bacteria that can reproduce very, very quickly, usually the rod-shaped ones, very, very quickly. Smaller size. For an example of some of this PGPR, you have the sidiophores, and what these do is they send out enzymes onto the pathogen that prevent it from absorbing, or let's just call it absorbing, iron. Iron being a very crucial part of life. We all need iron, right? What happens when we don't need iron? We get very, very sick and our organs shut down very, very quickly. These pathogens, microorganisms, are no different. So these specific PGPRs, talk about this sidiophores, are able to block iron absorption on pathogens. It's kind of like a lock and key good way to just take anybody out. I mean, anybody. So if there's a pathogen or something that's affecting the plant, it will send out these sidiophores. Also, these sidiophores exist just free living in the soil as well. There's a wonderful professor up at UH Hilo that is studying these. His name's Dr. Oeya, very, very nice guy. Okay? Let's talk about biofertilization. Is another one of those direct effect of beneficial microorganisms. Well, of course, we have biofertilization. What does that mean? The fertilization in terms of what? Say it.
The environment.
Speak up. No. The plant, correct. So, biofertilization, bio meaning what? Biology. Life. So it's not really the environment, it's more like the creatures that are in the environment. That's the biology, right? So if you're not talking about the rocks and you're not talking about the twigs, you're talking about the microorganisms only.
Within
So-
the ecosystem.
So yeah, the biofertilization is fertilization of plants by organisms. So we have non-symbiotic nitrogen fixers, exophytic nitrogen fixers. These are ones that live on the outside of the plant and create nitrogen for their own good, and just so happens that some of that might benefit the plant as well, because they might produce more and it dribbles out the corner of their mouth and there it is, ready for the plant. Well, sort of. A lot of these non-symbiotic nitrogen fixers seem to be really close to the root zone when we do kind of observations of that. So it's hard to say if it's non-symbiotic. They're certainly not mutualistic, or they don't have this close relationship, but you can tell that a lot of these nitrogen fixers colonize around root zones, and that's just a fact. It's probably because there's a lot of nutrients there, and they're doing their own thing. They're taking nitrogen from the environment, but they need a certain nutrient that comes from a plant. Okay, so they're not doing it to benefit the plant, but they might need the plant for the nutrient source. So they stick around pretty close to the plant, but they're not inside. Got it?
So the flower is what?
I'm
So
sorry?
that, can you, like, what's in the picture?
Yes, this is a non-symbiotic nitrogen-fixing bacteria. This is what it's a species, I don't know the name.
But that's what they look
that
like,
is.
right?
Well, they look like all kinds, yeah. Increased availability of phosphates. We learned that phosphorus needs to go through microbial metabolic activities to be plant available in our plant fertility class, in the soil fertility class. So when you have biofertilization, when you have microorganisms breaking down these phosphorus containing materials, eventually you have plant uptakeable phosphorus, which benefits the plant. Release of specific nutrients. Some microorganisms will release specific nutrients to a plant if a plant needs it. How does it know? The plant sends out an exudate that will cause sulfur releasing bacteria around the root zone. A specific exudate that will, when it sends it out, only those sulfur digesting bacteria will come and they will release sulfur into the environment, and then the plant will take it up. Got to remember that the plant is in complete control of the rhizosphere. It knows what it's doing, it's sending out things at certain times, it knows it has an electrical charge, it's changing the charge here, it's changing concentrations here, it's changing pHs here. It is in complete control until it gets out of control. Right? Got that? Release of specific nutrients. Let's talk a little bit about induced systemic resistance. It's a little different than systemic acquired resistance, right, Gino?
Yep.
And systemic acquired resistance, which I didn't really get into because this is more of, like I was telling you earlier, the microbial side of this study. And what it is when microbes activate a defense response in plants despite being non-pathogenic. So, a biotic factor, microorganism comes in, goes into the roots, the plant freaks out, this thing's in me. It sends ethylene and jasmonate, which is another hormone, jolting up the plant out here and all the way to the tips. And what does that do, this specific hormone, and then in addition to this one, it triggers defense mechanisms on the plant. So whatever the defense tools that plant has in the tool belt, it sends guns blazing. Nobody touch me, nobody get near me, I'm gonna get you. And then, but it just so happens that this bacteria really didn't want to hurt anybody. And then the plant realizes it's not a pathogen, it's like, all right, but I'm still watching anybody. That's kind of what it is, right? It's looking around. Yeah, I know somebody's coming, I'm all heightened. The plant's all heightened, ready to go, puffing its chest out, right? So it triggers signaling pathways within the plant cells that lead to higher pathogen resistance in the host. Bacillus family is very commonly going to be a trigger for ISR. And you got to remember a lot of those are pathogens as well. So you can't, just know that that specific family is usually responsible for the ISR. Induced systemic tolerance is for abiotic factors, meaning nothing alive, so lack of water or that kind of thing. So sometimes plants will get induced systemic tolerance if they went dry once. They will be heightened, ready to go. So you can stress a plant out on purpose at certain phases of its life to get a certain return in the yield or product. ISR and IST are genetically co-regulated. So within the genes of the plant, it is written on the wall what happens if this microorganism comes in. We go through this freak-out phase. And then if this one comes in, we go through a different freak-out phase. Right? And then if we run out of water, what if it runs out of water and all of a sudden this guy gets in us at the same time? All right, so they have this co-regulated kind of talking back and forth. Right? Right?
What's the benefit for the elicitor, other than the plant is healthy?
It's not really too much. Yeah, it's just a healthy host for the most part. I don't really know. We don't know, I guess, enough about it to say that there is a kind of motive there. You know, it's more like he came along, and then this plant freaked out, and then now this house just got nicer. You know, kind of thing. Yeah. It's tough to say because we've only been looking at it for about 20 years. People went nuts in the 90s looking at that. Now we have inoculants that you can put on your plant that cause this reaction in specific plants. So corn might have a specific microorganism that does this freak out about. And so they tell you to spray your plant. It freaks out and becomes super strong He-Man style corn. You get huge ears and it's tough and it has no disease problems. So systemic acquired resistance and the ISR as well is very important to farmers. So if you're an agriculturist, understanding that concept is very important. And know that we've looked at pretty much every crop and figured out that, you know, these do exist and you can find out which ones they are and then you can inoculate your field with them. Okay, let's talk a little bit about types of beneficial microbes. Talk about 4 or 5 or 6 of them and then we'll take a break. First one I like to talk about is the Azospirillum. As you guys know, I don't do well in Latin. I didn't go to Catholic school, so I'm going to stutter through these as much as I possibly can. But the Azospirillum, being plant beneficial, I know it pretty well because it is a free-living nitrogen fixer. And these are the two species that are most prevalently known as plant beneficial. So if you're going to find them outside the plant, most likely it's going to be these 2 species. Free living, outside the plant. Here is a wonderful fish technology scan of a root. Can you see the cortical cells? Right? And it's kind of, this is like a root tip, right? And what is that? Purple, glowing purple stuff. It's actually this one. So it actually lives free living, but boy, that's awfully close to the root zone. I mean, if that root tip is blown up that size, you wouldn't visually be able to see the root tip, more or less know that there's any space between the two. It's a very close relationship, but free living meaning outside of the plant. The Bacillus family, one of my favies. You made it. Hi, welcome. We got handouts around. It looks like the kids went through everything. We got the Bacillus family. We're talking about beneficial microorganisms right now, and types of them. We're about to wrap up beneficials. Bacillus family is very, very commonly found in the soil in that it's probably the most prevalent in the soil. It is a bacteria. If you'll notice, most of the beneficial microorganisms are bacteria. Bacillus, some bacillus are pathogenic. Some of them can kill you as a human being, but there are plenty of plant beneficial bacillus species, including the subtilis right here. And does anybody recognize this one? What if I said BT? right? What is BT? BT is a bacteria that is being put into the cells of corn and other GMOs. These are the crazy poison fruits that you keep hearing about. Well, what they're doing is they're putting this BT into the cell structure, and so when insects bite the corn, they die. They say it has absolutely no effect on us as humans because this is a microorganism that as soon as it hits our saliva, or more or less anything beyond our lips, it will be dead.
Lovely.
It's a tough argument. Seems rational enough to me. You think that any of these bacillus are going to survive the digestive system? Probably not. Can't think of it as a poison. It's not a poison. It's a microorganism that kills insects when they bite that microorganism. So, it's a very, very useful, awesome, incredibly just amazing microorganism that should be used in farming, organic farming, wildly. But maybe not put into the genes of the corn. That's a little freaky. But I use this. I use it as an inoculant to put it out in the field on my fruit, on my vegetables, and it is a wonderful antibacterial, as well as a insect deterrent. I mean, it has lots of different qualities about it. It's good stuff. Okay, so don't hate on BT, right, guys? We got pictures right here. A is going to be a treated salad with Bacillus subtilis, and this is untreated. So we have salad. They call it salad, lettuce, Europeans, right? So we have a root zone, root hairs, and most of this here is going to be hyphae, right? These are fungi. They're totally attacking the root zone. And then here we have inoculated. So it has these like large gloppy cells that go around the roots and protect it from any fungi entering the root zone. And then here is, I think, tomato. This is inoculated and not inoculated. So it'll actually wrap around the root hairs as well. It's like a barrier, it's a border, it's a wonderful, wonderful, wonderful microorganism that everybody should be using on their farm. You can also get cultures of it and make them yourself at home, so you don't have to keep buying it. You just have to know how to do that. I'm not going to tell you how to do that, because that would be a 4-hour class. Okay, so we also have Pseudomonas. Pseudomonas. For the most part, if I'm listing it as a beneficial plant microorganism, chances are a lot of the species in that family are human pathogens. So there's a lot of human pathogens that are Pseudomonas. But there are several that are plant beneficial. They are biological control agents and PGPRs, as we talked about earlier. They increase the entire fitness of the host plant. Overall health increased by the use of Pseudomonas. One of the most commonly used would be the Pseudomonas fluorescens, which actually lives on the inside of the plant. Here is that fish technology, and we have, this is roots, this is fruits, this is leaves, and this is also fruits, like the side cut of a fruit. So this is actually living on the inside cells, or the inside structures of your crop. Just protecting it, doing biological control, watching hormone levels. Okay, Pseudomonas?
So it can allow the nutrients to come through, or is it protecting it, like it's sealing completely? and
These
it's
guys are a
going
little different than what you're talking about. What you're talking about is like probably more like these guys. So these guys will prevent
any-
things from coming in. Generally speaking, the Bacillus subtilis will just kind of completely coat the root zone, right? It goes away fast. because the plant eventually says, I don't want you on me, I don't want you on me, I want to have conversations with everybody out there and you're getting in my way.
Right.
But for the most part, it's protecting for that time period. So if you're getting heavy rains, if you're getting, instances where you're getting powdery mildew and like fungi is forming in the areas around you, maybe you should inoculate it with Bacillus subtilis. It is a preventative measure. If you are already inoculated with a fungi, Bacillus subtilis really only has an effect if that fungi is on the outside of the plant. Okay? So these guys, Florensins, on the inside, and this one down here, the Pseudomonas burrita, I mentioned it on the last class. We talked a little bit about soil microorganisms, so it's a heavy decomposer as well. So like these have a lot of different functions. They're not just out there to do these specific things. They do a lot of different things. Rhizobium. Rhizobium is a bacteria that has an endosymbiotic nitrogen-fixing relationship with legumes, typically. So what did I just say right there? Well, it's a bacteria, we know that. Endosymbiotic, what does that mean? Inside.
Relationship.
Yep, mutualistic relationship with the plant, and it does what? Nitrogen fixes. So it's able to take nitrogen gas, which it's given to by the plant to transfer it into nitrogen, plant-uptakeable nitrogen, right?
So you would see that in like soybeans?
Yes, soybeans, you see it in koichi, you see it in pretty much every legume, you see it in perennial peanut. They all got it. They all have the ability to do it. They don't all have the bacteria. It's common for them to have the bacteria naturally in the soil. But sometimes you will find that if you're over-farmed or industrial farming type of thing, you don't have these bacteria. So the poor soybean farmers in the Midwest, they have to inoculate their field with these microorganisms in addition to putting out seed fertilizer and all these crazy chemicals and all that. So it's like, how do you do it? Well, what they do is they actually don't inoculate the field, they inoculate the seed. They put the microorganism on the outside of the seed, so when the seed is plopped in the ground, it already has established a relationship with the plant. Okay? So this is a really great cross-section SEM scan of one of those nodules that you'll find on a legume root. And these are all tiny little bacteria, look at spaghetti, they're all packed in there really super tight. Why are they packed in there super tight? Well, there is no airspace because there is no air in a nodule. You can't have nitrogen fixation in an aerobic condition. It has to be in an anaerobic condition, no air. Which is why when free-living nitrogen microorganisms, nitrogen-fixing microorganisms fix nitrogen, they do it on the inside of their cell structure where there is no air. Okay? Well, there's two kinds of mycorrhizae. We'll detail those real quick. Mycorrhizae being typically one of the only fungi that have a beneficial relationship with plants. So there's not too many fungus out there that have a relationship that benefit plants and benefit the fungi at the same time. They're usually going to be the pathogen. So bacteria, very heavy on the beneficial side and the decomposing side as well. Two kinds of mycorrhizae. There's endomycorrhiza. Of course, mycorrhiza. Endo meaning what? Inside mycorrhizae. Of course, it's a fungi. Endosymbiotic on the inside, mutualistic soil fungi. It's also called arbuscular mycorrhizae. So some people even just shorten it to AM. So it's pretty commonly known in the agriculture industry as AM or arbuscular mycorrhizae. Endomycorrhizae is also what it's called. Here's a great example of this mycorrhizae, Glomeromycota. Here is a root root. The blue. The yellow is hyphae from a mycorrhizae. This hyphae extends out of the root zone. You can't barely see it. If I turn the lights off, you can see it. Can you see this?
Yeah.
Okay. So it extends out of the root zone and into the soil. And what is it doing? It is gathering water and nutrients, putting it into its cells, pumping it through their cell structure all the way back to the plant. They are giving those nutrients to the plant, and only the nutrients the plant is asking for, and absorbs the nutrients. And in exchange, it gives the fungi whatever food source it desires. At the time, it might want heavy sugars today. Today I need a lot of water. I'm thirsty. I had a lot of sugar yesterday. You know, whatever. And so there's this conversation on both sides. What I need, what do you need, what do I need, what do you need? Don't go out and get any more zinc. I have plenty of zinc. Thank you very much. We need some nitrogen. You bring that up? Appreciate it. I'll give you some sugar. And that sounds bad, didn't it? So yeah, it's that kind of relationship. And then of course you have, why is it called endo, right? Very, very, very important why it's called endo. Is because it actually goes inside the cell of the root. Okay? It penetrates on the inside of the root. You have ectomycorrhizae, ectosymbiotic. Typically, it's the type of mycorrhizae relationship you find on woody plants, usually trees. Why? It's because it's kind of hard, right? I mean, poking into the cell of a tree versus into the cell of a corn plant is a completely different demon. So, trees tend to have this endomycorrhizae relationship where here's, for instance, if there was no mycorrhizae, these colors represent different kinds of beneficial bacteria and nutrients, right? And then, of course, if you do have mycorrhizae, you have all these filamentous cells that branch out and act like root hairs, and they bring in all this wonderful good stuff from the environment and brings it to you as the plant. What is it increasing? Surface area of your roots. That's the key to mycorrhizae. Increase the surface area of your roots, and there's that wonderful relationship. So these are two of the ectomycorrhizae that have relationships with trees in Hawaii, actually all over the world. So there's only a small amount of population of plants that don't have that relationship.
So if I want to get more fungi for my fruit trees, I would go somewhere where there's fruit trees and maybe the soil, under the soil a little bit to get some of that.
Yeah. I mean, you're basically going to inoculate from another area,
Mhm.
sure. Yeah. Just be sure that you're pulling it from the healthiest tree on your lot, because if you're bringing fusarium from one tree to another, then you're. So be really careful. So just look at your plant, know it's healthy, look for galls, look for cankers, look for necrosis, all these wonderful things that I'll get to in the next section. Okay, let's take a break, talk about plant parasites, yeah. Be back here in about 10 minutes.